Right. I'm going to discuss MCQ questions of 2022 paper. Now, I have discussed several questions. Today, I am starting to discuss the question from question number 43. Let's look what is the question number 43. This is the question. Select the correct statements regarding clotting of blood in man. First choice is fibrinogen is converted to fibrin by thrombin. Second one, prothrombin present in platelet is converted to thrombin. Heparin prevents conversion of prothrombin to thrombin. Vitamin K is a clotting factor present in blood plasma. Platelet becomes sticky due to agglutinogen present on their surface. Okay, we take one at a time, and also before that, we need an overview of a blood clotting process. So let's take the first choice, and also we try to understand what is a blood clotting process. Right. Number one, if a blood vessel is like this, when there is a damage to blood vessel, then the surrounding tissues damage. As a result, there is a gap here and the blood can leak out from the blood vessels. Okay. So from this area, blood can leak out. This is called bleeding. Immediately, within a few seconds, the blood clotting process starts. So blood clotting process is to make a seal in this gap and prevent further loss of blood, which is very important to prevent loss of blood, and also important in order to prevent entry of pathogen from this area. Right. Now, first step is the exposed tissues. So we know the blood vessel is made out of different tissues. There are different layers. Inner layer is the endothelial cells. The inner layer is the endothelial cells. That means it has a cell like this. And then underneath the endothelial cells, there are different tissues, and these tissues depend on the blood vessels. So these tissues depend on the blood vessels. When we considering bleeding from the vein, because veins are mostly on the surface level, we can also find certain structures like collagen. So this is a collagen fiber. Okay, now the speciality of collagen is it has a special site, and this special site is a receptor for platelet. Then next event is platelet. This is a platelet, and the platelet exposed a binding site for the collagen. Platelet, and this is the receptor, and this is a binding site. I just write here receptor, and the other one is a binding site on collagen. As soon as platelet bound to the collagen, then a process called activation take place. During activation, what we can observe in platelet change the shape So it's a round shape, and now platelet start to change a little bit like a star shape. Not only that, platelet secrete chemicals. Platelets start to secrete chemicals. Right now, the first event of clotting is the inactive protein in the plasma. So this is a plasma. And there is a protein. The protein is called prothrombin. So prothrombin is in a inactive form. So it doesn't do anything when it is in the inactive form. This chemical released by platelet can activate prothrombin into thrombin. This is prothrombin and this is thrombin. Thrombin is an active form. So when it active, it can further convert a certain other component present in the plasma, such as a fibrinogen. So fibrinogen also in an inactive form. So when it comes to a active form, it makes fibrin. So this one is a fibrinogen, and this one is a fibrin. So fibrin start to join together to a insoluble fibrin structure. So this fibrin act as a net mesh, and then it can further trap the cells, platelet, white blood cell, red blood cells, and it can form a clot in this place. Okay, certain other things also require this process. For example, production of prothrombin, vitamin K is important. Vitamin K is important. Production of prothrombin. It is not directly involved in the clotting process. So clotting process is separate process, but vitamin K is important to production of prothrombin. Vitamin K helps to produce a prothrombin. Okay, after that, when we take the platelet, so this platelet can also join together like this. So they can aggregate, and also they can trap in this fibrin mesh like this. The blue color one is a fibrin mesh, and the aggregation of platelet. So this clot represent platelet aggregated, and the net, which is fibrin, serve as a net to trap this platelet. Then the platelet aggregate. Other cells can also aggregate, for example, red blood cell, and for example, white blood cells. They can also aggregate, and then they can make this clot. Okay, fibrinogen is converted to fibrin by thrombin. Yes, this is exactly correct, because this one is a fibrinogen, which is inactive. It's converted by fibrin by thrombin. Thrombin is an enzyme. So when it activates, it's act as an enzyme and convert fibrinogen to fibrin. So first one is correct. Next one, prothrombin present in platelet is converted to thrombin. No, prothrombin present in plasma. So I clearly said yellow color area is a plasma. Prothrombin is is an inactive protein which is present in the plasma. As a result, this one is not correct. Okay, next, completely different statement. Heparin prevents conversion of prothrombin to thrombin. Right. Let's say, what is this sentence? Now, what is heparin? Heparin is used as anticoagulant. What is anticoagulant? Prevent or prolong blood clotting. Right. This heparin is a medicine which is used in the clinic. For example, certain cases, if a person is undergoing a surgery, after the surgery, many tissues are damaged, and there is a chance of making blood clot. So after patient recovered from the surgery, still there is a chance of making the blood clot. For example, if we understand his blood vessels connected at suture. Let's see, this is the surgery done. There are two blood vessels connected with each other. For that, we use the sutures. But inside, there is a still damage. When the blood is flowing from this place, so because of this damage at this area, there's a chance of formation of clot. If the clot form, this is dangerous because this can block blood vessel in the heart, it can block blood vessel in the lungs, it can block blood vessel in the brain-like places, and this is a life-threatening situation. As a result, these patients are prescribed to take heparin. Heparin prevent uh, conversion of prothrombin into thrombin. I mean, this is done by enzyme. This is done by clotting factors. Heparin inhibit these activation factors which can change prothrombin to thrombin. As a result, blood clotting is prolonged or blood clotting is not happen. Then that statement is also correct. So A and C correct. Vitamin K is clotting factor present in blood plasma. No, I told you it is not a clotting factor. I separated vitamin K from this clotting pathway. It has indirect effect production of prothrombin. It doesn't act as a clotting factor. So that case, this statement is also incorrect. Last one, platelet becomes sticky due to agglutinogen present on their surface. If I mention this one, platelet aggregate, that means they become sticky because they can aggregate with each other. Right. Now, the problem is if you try to focus this word, where have you seen this word in the resource book? Can you remember where did you see this word in the resource book? Okay. You have heard similar word in the resource book called agglutinin. agglutinin. Now, where did you see this agglutinin? If you take a red blood cell, and the plasma, and we discuss about the blood groups. So when you're discussing about the blood group A, B, AB, and O, what we mention for each blood group, in some people, they have a different type of antibodies. For example, I take one example. If I take blood group B, so blood group B, the person have this person has a special glycoprotein called B glycoprotein. B glycoprotein. Now the B glycoprotein serve as a site for antibody binding. This is an antibody. But this person, antibody is A. Antibody is A. It does not bind with B. So there is no chance of binding. Right? So the blood antibody A does not bind. But if you take another blood group, let's say blood group A, this is a red blood cell, and similarly, they also have glycoprotein A. But this person has antibody B. So antibody A and antibody B are specific for anti-A, and this one is an anti-B. Now, if there's a transfusion happen, this person, let's say transfusion A to B, now A get red blood cell from B get red blood cell from A. Now that's a red blood cell, and it is A. Now this time B has A protein. Now this is chance this antibody to bind. Anti-A can bind to the glycoprotein A. After that, red blood cell lies because of antibody and complement protein mediated reaction. Red blood cell burst. However, you haven't learned the word agglutinogen. Okay, but you learned the word agglutinin. Right. Now, what we actually refer in agglutinin is this antibody. Agglutinogen is this glycoprotein. So that's a glycoprotein on red blood cell, agglutinogen. So the last choice is correct up to this level. Platelet becomes sticky. Up to that, this is correct. And this part does not refer to platelet. So agglutinogen is the word typically used for red blood cell. Red blood cell is the one we used which has the ability to make an immune response by binding this agglutinin. So it's typically we use for red blood cell, but we can also use any foreign cell or any pathogen if they have some kind of glycoprotein on surface that can trigger immune response by bringing this agglutinin. Then we can call agglutinogen. And also one of the function of antibody is agglutination. That means antibody can bind with bacteria or any other pathogen from this agglutinogen and agglutinin, and then it can make a clump, a collection of bacteria which make easy to phagocytosis. So agglutinogen is the word we use typically when it comes to red blood cell. So this part is not correct. So in this 43 question, it is only A and C is the correct answer. When A and C is the correct answer, we have to go for other option, option 5. So this question is very important questions. Question number 40 to 50. And one can argue correctly this agglutinogen is not refers to platelet, and there is no evidence and there's no documentations published research article even to say that platelet has agglutinogen. So agglutinogen is not the word used. It used in standard textbook aggregation, platelet aggregate. Okay, I will continue rest of the 40 questions.